Good morning. In the journal club discussion, we are going to discuss an article with the title Loss of tight-junction, loss of tricellular tight-junction tricellulin leads to hyposalivation in Sjogren syndrome. So we have been discussing about Sjogren syndrome, which has a clinical symptom of hyposalivation. Here in this article, we are going to discuss about the loss of tricellular tight-junction, that is in tricellulin. So let me just tell about Tricellulin. So this is the acinar cell, and we have the cells here. This is the tight junction here, and the tricellulin is the protein which is forming this tight junction. When there is an autoimmune disorder, there is interferon gamma which is secreted, which causes the STAT pathway to go, and it forms a miRNA which is 145. This miR145 goes and binds with this tricellulin gene at the 3' UTR region, which will prevent the formation of the tricellulin protein. This tricellulin protein, if it is not formed properly, the tight junction will not be formed, and which causes the macromolecular transport outside. So what happens? There is loss of macromolecules out in the saliva. So the salivary secretion becomes very thick, and that leads to hyposalivation. So this is the title. So loss of tricellular tight junction tricellulin leads to hyposalivation Sjogren syndrome. This is the hypothesis of this study. This article was published in the journal International Journal of Oral Science. So in the year 2025, with an impact factor of 12.2. So this was done in the Department of Physiology and Pathophysiology in China, Beijing, China. In the year 2025, this article was published. So I have explained about the tricellulin junction. So the novelty of this study is recent investigation suggests that correlation between lymphocytic infiltration and salivary gland dysfunction in the Sjögren syndrome patients is less pronounced than the previously perceived. So they are studying about the lymphocytic infiltration. They see that it is less pronounced as previously thought. So this observation underscores the importance of exploring therapies that enhance the glandular epithelial function in the context of Sjögren syndrome management. So marked elevation of macromolecules such as autoantibodies, inflammatory factors and proteins are seen in the saliva of SS patients, underscores the potential role of tricellulin in mediating this pathological secretory pattern which remains unexplored. So this study aimed to explore the role and regulatory mechanism of tricellulin in the development of salivary gland hyperfunction in Sjögren syndrome. So here they used human salivary gland tissue. They collected female patients because middle-aged and elderly women are most commonly affected group. So the lip salivary gland biopsies were obtained from 6 female patients with the Sjögren syndrome, and one therapeutic gland biopsy was obtained from another female patient of the Sjögren syndrome. Both the lip and the parotid gland patients who underwent mucosal resection were conformed to pathology. They were the controls. So the normal people were the salivary gland which is biopsied from the during mucosal resection, and the other people were from the Sjögren syndrome group, who were mostly the middle-aged women. And they did animal model saliva collection also. They used female NODs, non-obese diabetic mice. Because female mice have higher prevalence of spontaneous inflammation of salivary glands and are more prone to dry mouth compared to male mice. So 7, 14 and 21-year-old NOD mice and age-matched BA mice for control were purchased, and their blood samples and the saliva were collected. The salivary gland acinar cell specific tricellulin conditional knockout mice were generated, and they also generated this knockout mice for tricellulin. So histological and immunofluorescence staining were done. They did a qPCR. They found out what are all the RNA changes happening, and the Western blot was done to see the protein changes. ELISA for the level of albumin in the saliva harvested from mice was measured, and TEM was used to find the distances between neighboring tight junctions using the imaging software. Cell culture, the rat SMG epithelial polarized cell line and SMG C6 human embryonic kidney cell line 293T were used. So they used two cell lines, one is SMG-C6, one is 293T. Proteomic analysis were done. Proteomes of 3 cell line cell samples after interferon administration taken both in the control as well as the this cell line. And RNA sequencing, 4 SMG cell samples treated with the interferon for 24 hours and 4 control samples were used for RNA sequencing. And they used a trans-epithelial electrical resistance measurement. Here the cells were grown to form a confluent monolayer and the PER was measured at 37 degrees C using an epithelial voltmeter. So here the value reached a plateau when the TJ barrier integrity was well established among cells, indicating a maximum volumes and readiness for further experiments. So they wanted to confirm whether these cells they form the tight junctions. For that they used this trans-epithelial electrical resistance to confirm whether the tight junctions are formed. And paracellular permeability assay was used to visualize the flux of macromolecules across the monolayers of these cell lines. Oligonucleotide transfection was done, ductal dual luciferase report assay was done, and the statistical analysis was done to find the all the significant changes. And the results showed that the expression of tricellulin is reduced in salivary gland secretions. So they confirmed that tricellulin is reduced. And expression of tricellulin is decreased in hypofunctional SMG in mice also. So this was in human, this was in mice. And deficiency of tricellulin leads to hyposalivation leakage of macromolecules. That also they found. Expression of tricellulin is downregulated in interferon and SMG cells. So when interferon is gamma is administered, the expression of tricellulin is reduced in the SMG6 cells. And JAK-STAT MIR145 axis mediates the effect of interferon gamma on tricellulin expression. So they also found out which pathway is affected due to this in this process. An enhancement of TJ barrier function and restoration of tricellulin expression ameliorates hyposalivation in NOD mice. So they found out that this TJ barrier function causes the hyposalivation in NOD mice. So in conclusion, they found that the crucial role of epithelial tricellulin in maintaining the barrier function and its dysfunction in pathogenesis of cells. And consequently, targeting acinar epithelial tricellulin represents a promising therapeutic strategy for addressing the hyposecretion and macromolecule leakage. So they use two compounds, which is the AT1001 and MIR145 antagonist, which is used to inhibit the pathway where the MIR145 is formed, which is the main reason for the degradation of tricellulin protein. So this treatment substantially alleviated the hyposecretion and reduced macromolecule leakage in NOD mice. So when we see where the AT101 acts and where the MIR inhibition also happens, MIR antagonist inhibition also happens. Here it prevents the tricellulin depletion and here it inhibits the MIR145 to prevent the tricellulin degradation. So they are trying to maintain this tricellulin protein to prevent the loss of the tight junction, which will prevent the loss of the macromolecules in the saliva of excretions. So limitations, they used limited human sample size and diversity. They used a few labile salivary glands. The study was subject to were all females and middle age. So it was generalizability to male patients is not possible here. And the model system constraints and species differences. So the in vivo experiments on the NOD mouse model was used. And in cell culture, SMG6 rat salivary epithelial line was used. The aquaporin by credit ER model used for acinar cell specific knockout also shows off-target recombination in the lung. So the aquaporin 5 is expressed in lung as well. And the mechanistic depth and therapeutic translatability limitations were there. While this pathway of JAK-STAT was proposed, other cross-talk pathways were not explored, and therapeutic interventions were tested only in the mice and not on human testing was done. So the future scope involved validation in human cohorts and translational studies, and further mechanistic dissection and identification of regulators, and preclinical and therapeutic development has to be done. So this study used the research protocol was approved by the Peking University, and all patients signed the informed consent, and experimental procedures were approved by the Ethics Committee of Animal Research, and the guide for the care and use of laboratory animals was followed.